Hello, and welcome to BioC 2021. You're at the package demo session for ATAC-seq QC, and we'll be starting shortly here. First, we'll have a few housekeeping items. So this session is going to be a pre-recorded video that our presenters Kai and Haibo have prepared. And I think it'll work most smoothly if we hold questions to the end, but you can please feel free to put questions in the chat or in the Q&A as you watch the video, and then we'll have an opportunity to address them at the end. So in that Q&A tab, you also have the ability to upvote questions. And I guess since we have a video, if you want to ask a question live, we'll do that at the end of the session here. While the video of this session will be uploaded shortly here after the session is completed, and if you want to follow along, Kai just posted in the chat a link to, I guess it's probably the vignette that they cover in the workshop here, as well as slides, so you can follow along. So with that, I'm pleased to welcome Kai Hu and Haibo Lui from UMass Medical School, who will be presenting today. They're both scientists with the Bioinformatics core at UMass, and their package has been in bioconductor for almost 5 years and has seen quite a bit of use in that time. So I'm sure we're going to learn a lot today in this package demo. So with that, take it away with the video, please, Hajira.
Thank you.
Okay, everyone, this is Kai. I'm a bioinformatics researcher here at the UMass Medical School. Today, I will get you through what ATAC-seq is and how to perform downstream analysis with the package called ATAC-seq QC. Before I get started, I want to acknowledge the core developers of the package, namely Haibo, Julie, and Jianhong. I also want to thank Nathan, Michelle, and Lucio for their contributions during the development of the package. Here is the agenda of the workshop. For the first part, I will begin with an introduction to the ATAC-seq technology, and then describe a common ATAC-seq experiment workflow. After that, I will talk about the best practices for each of the steps. For part 2, I will talk about the main features from the ATAC-seq QC package, and also demonstrate the usage with some demonstration plots. The last will be a Q&A session, where Haibo will join me to answer questions. Okay, in case you want to replicate the demonstration plots from this workshop, you can choose to download the Docker image, which contains all the software and test dataset. To this end, first you have to install Docker engine, and after that, you must download the Docker image using the Docker pull command. And then you must start the Docker container with the Docker run command. Here you can put in whatever passcode you want. And finally, you have to open your browser, type in this address, and log in with RStudio as username and the passcode you specified in the previous command. For more instructions, you can go to this GitHub repository. And now I'm gonna briefly show you how to run those codes. So basically, the first thing you have to do is to copy and paste this Docker pull commands. Type into your terminal. This command will download the image from the Docker Hub. Depending on the speed of your internet, it will take 10 to 30 minutes to download for the first time. And after that, you must initiate the Docker image with the Docker run commands. And this will start the container. And now you can see that the container is already started. After you see this, you can go to the browser and type in the localhost like this. Log in with RStudio and the password. After you sign in, you can see this RStudio interface. Now, in order to see whether this container is working properly, you can try to load some of the libraries. For example, the ATAC-seq QC. Now you can see that this package can be successfully loaded into the environment, meaning that the environment is working properly. Okay. Okay, let's get down to the real business. So what is ATAC-seq? It is basically a simple approach to detect the open chromatin region. In order to better understand the principles behind ATAC-seq technology, let's first review the DNA packaging mechanisms in eukaryotic cells. As we all know, in eukaryotic cells, the DNA are super long linear molecules, and in order for them to fit into the tiny nucleus, they have to go through multiple packaging steps. The first step is to wrap around the histone core to form the so-called nucleosome structure. Along the genome, the DNA packaging is not even. For example, as shown in this figure, some of the region, the DNA can form the nucleosome structure, and the nucleosomes are stacked tightly together, therefore prevent the binding of transcription factors. We call those regions the closed chromatin. In contrast, there are some other regions that are free of nucleosomes. Those regions are open accessible to transcription binding factors. Therefore, they are called open chromatin. Although the open chromatin only consists of about 5% of the entire genome, most of the transcription factors will bind to this region. Therefore, they play an essential role in the gene regulation process. Typically, the ultimate goal of the researchers would be to determine if there are any changes regarding the open chromatin profile in the treatment group versus in the control group. In order to profile the chromatin accessibility landscape, scientists have proposed many methods. For example, DNase-1 is one of the first adaptations for such purpose. It utilizes endonuclease to cut at the open chromatin region, and as a result, the fragment will be mapped to the open chromatin region. Similarly to DNase-seq, ATAC-seq is another technology, but instead of using endonuclease, it leverages a special chemical to cross-link and isolate the open chromatin region before sequencing. In contrast to DNase-seq and the ATAC-seq, MNase-seq is another technology which leverages an endoexonuclease, which can cut every region except those that are protected by the nucleosome. Therefore, the pattern is kind of orthogonal to those that are produced by DNase-seq or ATAC-seq. ATAC-seq, on the other hand, is a relatively new technology. It leverages a hypersensitive transposase called TM5 to cut at the open chromatin region. Because ATAC-seq requires fewer material and the preparation time is much shorter compared to the other experiment, more researchers are switching to ATAC-seq over the years. For the next slides, I'm going to demonstrate a common ATAC-seq experimental workflow. On the top panel, I summarized the major steps involved in a typical ATAC-seq workflow. First, you have to isolate cells and prepare the nuclei from the cell, and then incubate the nuclei with the TM5 transposes. Those transposes are preloaded with the sequencing adapter. Therefore, they can fragment and tag the reads at the same time. And after the tagmentation step, you also need to amplify your library with the PCR before sequencing. And after you receive the sequencing results, you can perform many downstream analysis, including quality control, peak calling, peak annotation, nucleosome positioning, as well as the footprinting of transcription factors, and many more. Here in the middle, I try to zoom in for the tagmentation step. So basically, on top, the yellow beans represents the TM5, that are preloaded with the sequencing adapter. The red scissor here represents the cut site of the enzyme. Here I try to classify the resulting fragments into 4 different categories based on where they come from along the genome. For example, as you can imagine, most of the fragment will come from the nucleosome-free region, as shown by the bottom 2 tracks. And some of the fragments come from the region that span 1 nucleosome, as shown by the first track. And there are fewer fragments that come from the region which span 2 or more nucleosomes, as shown by the second track. This will result to a very distinct fragment length distribution pattern for ATAC-seq data, which I will talk more in the following slides. Now you have the basic idea of what ATAC-seq is. Next, I'm gonna cover the best practices for each of the steps. And here, the major steps are listed in the green boxes. I also try to summarize the best practices for each of the steps with the dotted boxes. For example, starting from the cell, you must digest away the background DNA before you isolate the nucleus. And you can also choose to reduce the organelle DNA using ATAC-seq protocol. After you have the nuclei, before the tagmentation, you have to optimize the ratio of the TM5 versus the nuclear counts in order to prevent overdigestion or underdigestion. And after the incubation, depending on the amount of the DNA, you have to optimize the number of PCR cycles. And as for sequencing, we recommend using pair-end sequencing. And like I mentioned earlier, since most of the reads come from the nucleosome-free region, a read length of 40 to 100 base pairs should be sufficient in most cases. In terms of the read depths, if you are using human sample, we recommend at least 50 million reads. And if you are interested in detecting footprinting, we recommend using at least 200 million reads. Although the wet lab protocol is relatively straightforward and simple, to ensure the best results, there are multiple quality control steps. And I try to summarize those quality control steps in the red boxes. For example, you have to perform the nuclear QC step and the microscopy. And after the TM5 incubation, you have to perform gel electrophoresis to inspect the fragment set distribution and use qPCR to determine the number of PCR cycles for amplification. And like all other sequencing projects, after you receive the FASTQ file, you need to perform FASTQC to evaluate the sequencing quality. And for downstream analysis, you have to perform post-alignment QC and post-peak calling QC. For the post-alignment QC, we recommend using the package called ATAC-seq QC, which is also the focus of this workshop. And if you are interested in the post-peak calling QC, we do have another workshop from last year, which you can refer to. In these slides, I will try to summarize the best tools for each of the steps. Again, starting from the FASTQ, the first thing you have to do is to trim off the adapter sequence with the software called Chromatic or cat adapter before mapping to the genome with BOTA2 or BW map. And after you have the raw band file, you can choose to sort and remove organelle DNA with some tools. You can also label and remove the duplicates with Picard tool. In order to remove other artifacts, you probably need to write your own customized Python or R function. Once you have the filtered band file, you can then use Max2 to perform peak calling and associate the peaks with genes using package gpig-ano. For functional annotations, we recommend using the software Grid, ChipEnrich, or Behist. And for other downstream analysis, we recommend using NucleoTag for nucleosome positioning analysis and Tubius for footprinting analysis, and PK2 for regulatory network inference with paired ATAC data. Like I said, the focus of this workshop will be on the post-alignment QC with ATAC-seq QC package. Therefore, for the following slides, I will demonstrate the usage of 9 functions from the package as listed here. For the demonstration part, I will first talk about the motivation behind each of the function, and then demonstrate how to use those functions for plotting. To start with, for demonstration 1, we are gonna assess the mapping status with the function called BAMQC. This function is relatively straightforward. You have to input a solid BAM file with the duplicate labeled, and the output will be numerous mapping statistics, including the mapping rates, the duplication rates, the mapping quality, and so on. And of course, there are many alternative tools you can use for the same purpose, like SumTools or PK2s. Once you have the sequencing data, you may wonder if the sequencing is deep enough. In order to evaluate this, you can use the function called saturation plot. It basically downsamples your reads and try to plot the count of the call peaks against each of the downsampled reads. And if the curve flat out at a certain point, it basically means that the library is saturated at that point. You can also use the function called estimate lab complexity to evaluate the complexity of your library. For example, as shown at the bottom, the purple sample flat out even at very shallow sequencing depths, meaning that the complexity of this sample must be pretty low. As we discussed earlier, a good quality ATAC data will exhibit a distinct fragment set distribution pattern as shown on the left. Basically, as you can imagine, since most of the reads come from the nucleosome-free region, you can see a largest peak below the 100 base pair range. And there is also a smaller peak around the 200 base pair range, which represents the fragment that contain mononucleosome. There is an even smaller peak around the 400 base pair, which represents the fragment that contain dinucleosome, and so on and so forth. In contrast, for the poor quality data, you won't be able to see such distinct pattern. When you have samples from multiple conditions or many replicates, you can choose to plot the correlation of the samples with a function called plot correlation. For example, as you can see on the left, the replication 1 is quite different from all the other 3 replicates. You can choose to plot either PCA or heat map, and the correlation is calculated by the counts from the promoter regions. Demonstration 5 is about shifting the aligned reads. The reason why we need to shift the alignment is because during the TN5 tagmentation, there will be a 9 base pair overhang created at the 5' end. And in order to center the cleavage event, we have to shift the reads that map to the forward strand by +4 base pair, and shift the reads that map to the reverse strand by -5 base pair. And this feature can be achieved with the function shift G alignment list. Demonstration 6 is about splitting BAM files. Basically, the BAM files can be split into different bins. For example, BAM files can be split into bins that contain only nucleosome-free fragments, or mononucleosome fragments, or dinucleosome fragments, and so on. And then the reads from different bins can be used to visualize signals. The next demo is about plotting aggregate signals around transcription start sites. As we all know, most of the TSS are localized in the open chromatin region, which are flanked by well-positioned nucleosomes, as shown by the nucleosome density plot shown here. Therefore, we can imagine for a good quality ATAC-seq data, the nucleosome-free fragment will be enriched in the center of the TSS, whereas the nucleosome-bound fragment will be enriched flanking the center of the TSS. In contrast, for poor quality data, you won't be able to see such enrichment, as shown on the right. This heat map can be created with the function called feature-align heat map. You can use another function called feature-align distribution to quantitatively visualize the signal density around the TSS. These two plots basically represent the same information from the previous heat map plot. So again, as you can tell, the nucleosome-free fragments are enriched around the center of the TSS, whereas the nucleosome-bound fragments are enriched flanking the TSS. In addition to the visualization of signals around the TSS, you may also want to visualize the signals across a broader range of genome. To this end, we will find the function IGV snapshot very helpful. In order to run this function, you have to have IGV available in your computer, and as you can tell from this plot, for good quality data, the signals will be enriched in the open chromatin region, whereas for poor quality data, the signals will distribute across the entire genome, as shown by the lasso track here. The last demo is about assessing the footprints of transcription binding factors. This can be achieved with the function called factor footprint. The idea is that the binding of the transcription factor to the open chromatin will prevent the digestion of the TM5 during the tagmentation step. Therefore, it will create a dent in the middle of the peak. For good quality data, the valley will be very deep, whereas for poor quality data, the valley will be shallow or even invisible. Now I will demonstrate how to generate those plots with RStudio. And for this part, I have prepared two datasets. One is of good quality, which is from the first ATAC paper, which is published in 2013. And another is of poor quality, which is from a unpublished dataset. We have performed all the pre-processing steps, and what you have to do is to download the Docker image, and then you are good to go. Okay, for the demonstration, I prepared several files that you can refer to. All the plots can be generated with the demo.r file. And I'm going to show you what the demo.html looks like. So first I can open the HTML file. As you can see, there are 9 demonstrations in this file. You can browse through each of the demonstration. You can see the code, the results, and some comments about the results. Okay, now let's go back to the RStudio. So for the demonstration, first you have to open the demo.r file by clicking this. And for the demonstration, first you have to load all the packages into the memory. And after that, for demonstration 1, we're going to use the function called BAMQC to evaluate the quality of the BAM file. To this end, first you have to specify the path to the BAM file, and then feed these BAM files into the BAMQC function. This function may take a little bit longer to run, especially when the BAM file is big. We just have to be patient. And after several seconds, you will see the results from the console, as shown here. For demonstration 2, we're going to use the function called estimate lab complexity to evaluate the complexity of your library. Again, this may take a few seconds to run. And you can use another function called saturation plot to evaluate whether your sequencing is saturated or not. In order to run this function, you have to prepare a pig file which can be produced with the Max2 software. For demonstration 3, we're gonna assess the fragment size distribution. To this end, we leverage the function called fragSizeDistribute. And again, here for the good quality data, you can see the distinct pattern. Whereas for the poor quality data, you can only detect one giant peak below the 100 base pair range. For demonstration 4, we're gonna assess the similarity of the replicates. For this demonstration, we're gonna use the sample data from the package itself and use the plot correlation function to generate a heat map. As you can see, input 1 is quite different from all 3 other inputs. For demonstration 5, we're gonna shift the alignment using the function called shift galignment list. The resulting BAM file will be saved to the shiftedgood.bam or shifted_bad.com as specified by the outBAM parameter. This function may take a little bit longer to run, which you have to be patient. Okay, now demonstration 5 is finished. We can move on to split the BAM files into bins. And to this end, you can use the function called split galignment by cuts. The result will be saved into the outpath specified here. Again, this function may take a little bit longer. We also have to be patient. Okay, now all the BAM files are successfully split into different bins. Next, we're gonna plot the aggregate signals around the TSS using the function feature align heat map and feature align distribution. To this end, first we have to specify the specific BAM files that we want to work on, and also find the TSS coordinate information as well as the library size information. After we have all this information, we can then run the function called enriched fragment to calculate the signals. And also take a log scale of the signals. And finally, we can visualize the signals with heat map or the density distribution, like shown here. This is for the good quality data, and the similar thing for the bad quality data. Just run the same similar commands. And for demonstration 8, we're gonna leverage a function called IGV snapshot to streamline the IGV snapshots. And to run this command, you have to have RGB available. And because I don't have a RGB available here in the RStudio, so I comment this function out, but you can uncomment it and run it in your local computer. And the last demonstration is about assessing the DNA binding factors. For this function, first you have to get the motif information and use the function factor footprint to visualize the footprint of the transcription factor. Again, this may take a little bit longer to run, and we just have to be patient. Okay, now as you can see, the footprint plot has been generated on the right. Okay, now we can move on to the last session, which is about the QA. Here I recommend submit the questions to Bioconductor support site. There are two benefits. First of all, this website is monitored by the developers, so that the questions can be answered very quickly. And on the other hand, your questions can benefit other people as well. And now we're open to questions.
So it looks like we do have a few questions in the Q&A here, and so I think one question we had is from Adrian, who asks about expected library complexity.
So the library complexity, yeah. So the library complexity is kind of a relative matrix. So if you have multiple replicates for a given condition, so it's better to use the library complexity estimate to find the library which the replicate which has a higher complexity. Yeah, but it's hard to say. So what's the cutoff for a library complexity? So it's the library complexity also depends on the available of how many nuclei you have for the transposition process, and also the transposition reaction, how the ratio of TM5 transposase relative to the nuclei numbers. So yeah, so it's yeah.
Experiment specific. Is there a complexity where you would just say, give up and say like these data aren't worth analyzing, or does it really depend on the context of the experiment?
Yeah. For some situation, if you have very little bit of biomaterial, you cannot get a very, very high library complexity, but it's still okay to do the analysis. So, but in case you have multiple replicates, you have enough material, you can, by using the function called estimate library complexity to find out which replicates is more complex than others. Then you can use that library to do more sequencing to get enough data, so you can have a better discovery of the open chromatin regions.
Great, yeah, thanks. It looks like we've got another question about using max3, I guess, versus max2.
Yeah.
Calling, if there's anything, I suppose, that we need to.
So the max3 is very similar to max2. I just recently looked at their documentation, but they do some like a source code change, so they can run faster and more efficient. So to use the mask2 for the ATAC-seq peak calling, so it's better to use a treat the reads as a single-end reads when you do the peak calling, and use the shift and extend the size option to adjust the cutting size and to like shift the upstream for 75 base, then extend the 5' for 150 base. Then the cutting site is going to be the center of the fragment, which is a better representative of the open chromatin region, instead of use the paired and the bound file to do the peak calling. Since the nucleotides between the cutting sites may not representing accessible region. Only the cutting sites represent a region which is accessible to the TM5 transposase. So another better choice is using the gene reach, which is specifically have an option for the ATAC-seq data peak calling, and also can handle the multiple replicates and get a consensus peak instead of using MAX3, which can only handle each replicate. Then you have to merge the peaks from each replicate.
Great, thanks. So Beatrice asks about causes of bad quality plots in the frag distribution size function.
So distribution size function is the most important metric whether the library have a good quality or not. Yeah, so theoretically you should see like a periodical pattern for the nucleosome-free region, mononucleosome, dinucleosome, and even trinucleosome. So you should see more signal located in the nucleosome-free region, then followed by the mononucleosome region and the dinucleosome, and so on.
And if you don't see that, how would you?
Yeah, I do have one more comment.
So sometimes if people do a library selection, they can cause some bias for the distribution, the reads, the sequence data-based insert size distribution. Yeah, so if they didn't do any size selection and they see very weird, like only you see one of the peak, which is smaller than 100 base, that means you're adding too much TM5 transposase, and it's over transposed. Many of the regions that are not open, they are cut by the enzyme just because too much enzyme are added. So sometimes you may see only have a very small fragment of reads in the nucleosome-free peak region, so it's less than 100 base, but many other fragments are located in like a mononucleosome, dinucleosome. That means you didn't add enough TM5 transposase to the reaction, so that means that can affect the downstream open chromatin discovery, because many open chromatin regions that are like an enhancer, which are less open compared to the promoter. Then for those regions, if your library was not the distribution of your fragment you are expected, so you only have a very little bit of reads come from the open chromatin region. That means you have less chance to discover enhancer and like an insulator, something like that. Yeah.
Thanks. I had a question that, as I saw the presentation, I was wondering if there have been any large-scale efforts to generate benchmark data sets to understand the impact of QC and algorithm choices. Like in RNA-seq, we've got like the MAC-QC data set, and there have been some efforts for this also in like single-cell RNA-seq. are you aware of analogous efforts for this in ATAC-seq so we can get a better handle of what the impact is between algorithms or parameters? So I guess like the ATAC-QC, they took a bunch of standardized samples and sent them to various sequencing centers so you could get some handle on like the variability introduced by both like the sample as well as kind of the more batch level variables. In some of the single cell data sets like Matt Ritchie with his like SC mixology, they took various tissues and then combined them at known ratios in order to generate kind of known signal in the data set too, because you know that you're mixing like tissues A and B at a known ratio, so you should kind of see like the whatever effects you should be kind of like 50-50 between effects in A and B. And yeah, I was just wondering if there's anything like that for in ATAC-seq.
So you mean to like make some reference data, use that as a just like the profile of the reference data as a base to compare to the user's data?
Yeah, really a benchmarking for benchmarking algorithms as opposed to like benchmarking the biology. So if we run some known sample a bunch of times, then we'd hope that the QC that's doing a good thing would minimize the variability between these replicates. For instance, I mean, it sounds like maybe this doesn't exist yet with.
I don't know for this.
But yeah, well, someone just needs to write the grant and get the money to do it, right? So.
Mm-hmm.
So I don't see any other questions in the Q&A or chat. So if our speakers don't have any other final thoughts to add to this, then we might end a little bit early and we can all either get some dinner if you happen to be on the time zone where it's dinner time, or just a break before our next sessions that start in 15 or so minutes here. So yeah, thanks again to Kai and Haibo, and thanks for the great vignette, and I hope it will be of use for everyone.
Thank you, Andrew.
Thank you. Thank you, Andrew. Thank you all.
Yeah.